Welcome to Translating Proteomics, where we explore the science of proteomics and its growing impact on basic biological research, drug development, and more. And now your hosts, Parag Mallick and Andreas Huhmer of Nautilus Biotechnology.
Welcome back, everyone. I'm excited that today we're going to dive into applications of proteomics, both expected and unexpected. Andreas, you have been a pioneer in helping people make tremendous progress on a wide range of applications. Let's start. What is the favorite over your whole career? What is the favorite application you have of proteomics?
All right, that has to be connected to food. I'm a foodie like you. And so I was traveling to New Zealand and we were invited by a customer who was interested in studying milk.
Okay.
The reason was that they were producing high quality cheese and high quality milk products for the Asian market, particularly the Japanese market. And the feedback from customers was that, there was a particular cheese that was of such high quality that, the chefs in Japan would, fight over it. So the question was, how could you make more of that cheese? So to make this story short, they eventually studied those milk proteins, and it boiled down to the fact that very specific, very specific type of cows, and a very specific pasture where they were eating at a very specific time of the year, would create a very specific profile of casein and other milk proteins that would ultimately lead to this phenomenal cheese.
Wow.
So back then, obviously, it was very difficult to study those highly similar casein proteins. You appreciate that they're very complicated because they calcium binding, and so they create a lot of issues in a traditional proteomics tool space. But I can imagine that in this next 10, 20 years, we'll have tools where it becomes effortless.
Yeah.
To really measure, every batch of milk, and then, make the best cheese ever. So I have no problem with that. If proteomics can make better food, I'm all for it.
That's fantastic. Well, I think for me, my favorite application is a little closer to home. There was just a tremendous series of work by Matthias Selbach's lab where they really were able to, in detail, understand how transcriptome and proteome work together, how protein turnover drives regulation. And for me, it was really the first time that we started to understand how the rates of the transcriptome and the scale of the transcriptome, 4 orders of magnitude, were dwarfed by the variation in rates of turnover of proteins, which spanned on their own 8 orders of magnitude. And then the abundances spanned 8 to 10 orders of magnitude. And so I was just so excited to see that because it helped really explain this handshake for me, the handshake between the transcriptome and the proteome, and how the proteome is able to accomplish so much and so differently than the transcriptome.
I agree with you. I read the paper as well, and I think I was equally enthusiastic about it. I think it was a real key moment, particular as people always compare the transcriptome and the proteome, and wanted to be the transcriptome an equivalent measurement to the proteome measurement, and that paper really put the knife into that argument, right?
Yeah, that we really we shouldn't expect them to be correlated to each other because they have that relationship between them is so much of the regulation. And just beautiful, beautiful work. So, tell me a little bit about when you think about proteomics and you think about applications of proteomics, you've seen a tremendous range, but where are the places that you see proteomics having the biggest impact?
Well, proteins are literally everywhere, right? I mean, we mentioned plants a little bit. Mm-hmm. I think plant biology is highly neglected, not only from our funding agencies, but also in general in the way that biologists study it. Yet we all depend on plants because without plants we couldn't live.
Sure.
So I think there will be, if we indeed have ubiquitous, easy to use, highly, massively parallel measurements of proteins, I think we'll be, attacking problems that we have not attempted to attack now because we simply don't have the tool set for it. So to me, the plant space is vast. I would also say, areas such as, pests. I think we literally poison these, pests right now, but also poison the environment in the process. I'm pretty sure we can come up with much cleverer tools in the future, utilizing the knowledge of the proteome of whatever the pest might be, right? So I think that these are areas where I see tremendous transformation happening.
Yeah.
And then maybe the last space is that we can actually now learn how to build new types of proteins. I think to a certain point we're doing this for washing detergents.
Yes.
So there's fair amount of
right. Dow Chemical has a tremendous effort on new enzymes for cleaning clothes better.
Exactly, right. And most people probably don't realize when they wash their clothes, it's mostly proteins that clean up your clothes. But I think there is huge number of applications in where proteins can do all the work for it. So some of the areas that I have encountered before is when people were trying to, for example, make artificial skin. So to that, maybe 10 years ago, people were trying to grow, particular fibrinogen protein in tobacco plants. I think we could probably very soon make those in fermenters at massive scale, so we can actually create artificial, proteins that we need for wound healing, and that's the case.
Well, one of the largest applications of large scale fermenters right now for proteins is Impossible Burgers. And so I think while we don't think about it very much, I had a really fun conversation with the CEO of Impossible Foods, and he was really excited about studying the hamburger proteome and and emulating it, which is related to food, but a very different take on it. When I think about applications of proteomics, there are really three buckets for me. One is in biomedicine, of course, finding new therapeutics, proteins that are differentially expressed between healthy and diseased tissues. Number two, biomarkers, finding things that help us diagnose diseases earlier and type people better so that we can match therapeutics that are going to be effective to them. And I think also just the entire continuum of therapeutic development, mechanism of action studies, toxicity, et cetera, huge opportunities in proteomics. Beyond that, if we look at some of the global challenges we're facing today, we're facing challenges in, of course, global warming, climate change, and there are questions about what can we do from a perspective of engineering novel bacteria or novel plants that operate on fundamentally different systems that maybe can fix carbon more effectively. Can we build a better tree that is able to, its proteome, its enzymatic pathways are wired to absorb so much more carbon? Are there bacteria that we can engineer to convert nuclear waste to some other something less toxic? I think there's a term in that area of synthetic and systems biology for environmental impact. I think there's huge potential there. Also, with regards to our food supply chain, you mentioned some of these areas. We have aspects like drought tolerance and pest tolerance, and these are huge issues, as well as nutritional value. So are there ways that we can understand strains from their proteomic response profile to stress and say, you strain, your proteomic profile is estimating that you're going to be really drought tolerant, and do a better job of identifying those. So separate from genetic engineering, just measurement and metrology.
Yeah. Wouldn't it be great if you had a way to measure proteins that were independent of the existence of the genome, right? Because most tools now really need the reference of the genome, but you could imagine that you can study any protein from any protein, from any organism that actually produces those proteins without even having to have their genome in place, right? I remember a story where I was talking to a researcher in Malaysia. One of the agricultural products was export of shrimp, and at that point in time, and I'm sure it's changed by now, the genome of the shrimp was not sequenced. So that poor researcher had to use the next, relevant organism, which was the Drosophila fly. So
really, I wouldn't have expected that the Drosophila was the closest organism to a shrimp.
That's right. And so it was obviously very, very incredibly difficult to sort of interpret the data that she was getting either with mass spectrometry. She was trying to, scout some of the antibodies from other organisms. But she was making very, very little progress despite enormous efforts. Yeah. Because simply there was not the references there that she needed. And so, I hope we can, overcome these problems with, these next generation of tools, particularly in the proteomics space, where you don't depend on, making specific,
having a reference.
Having a reference, yeah.
Yeah, that's really interesting. So changing gears, we've talked about a number of incredibly practical things. So, and I would consider a next generation cheese an incredibly practical thing. Way out there, what is the craziest application that you can think of for these emerging tools?
I think when we hear about the story that we have basically created a synthetic genome that may give rise to a new organism, I think we could probably build subsystems of protein-based subsystems that would do work for us. I mean, we know that DNA can already be used in many cases to create nanostructures. I think we'll be able to do the same for proteins. However, I think what we're going to be capturing with proteins is much more the dynamic aspect of it. So I could imagine having a biological clock.
Oh, right? Interesting.
Fed by ATP. I mean, we have that internally, obviously, and plants have that. But maybe that's something that is maybe useful. Maybe just the curiosity, but creating a biological clock that can independently work. Maybe as a.
How would it deal with daylight savings time?
Yeah, I don't know.
It might have to change its media.
Although my understanding is that in the biological clock, there is actually a reset mechanism where you can actually reset the biological clock, right? And that obviously works when you're trying to, travel by plane to a different region of the world. Yeah. But I think there can be, I may not have the best example, but I think there's probably dynamic subsystems we can build that allow us to study, dynamic phenomena.
Yeah.
I mean, you could think of, a self-adapting pest.
Yes.
Right. That essentially senses that the pest is trying to kill has, evaded the defense mechanisms. And so now you change the attack, right? Yeah. And the biological system is completely able of adapting to that, to that new situation. I can imagine that.
Yeah. That's really interesting. So I as you know, but you all may not, I started in proteomics because I wanted to be an astronaut. And so I was always curious about how the proteome changes in response to zero gravity and how that led to changes in the calcium cycle or regulation that led to muscle degeneration in space and bone atrophy. So while it's not that far out there, because advances in understanding that might help us here on Earth of helping people live longer and healthier, for me, this new frontier of proteomics, it would be amazing to be able to bring it back full circle to when I first started in the field and was hoping that we'd be able to use these kinds of platforms to ask those kinds of questions.
So you envision a protein in space.
I envision a protein in space, and I envision studying astronauts extensively and possibly having to go to space in order to do these proteomic studies.
Oh.
NASA, if you're listening. So I think one last question for you. We've talked about how we're on the cusp of a major breakthrough in proteomics and a number of exciting applications. What is needed? What is the if we think about these applications and the next set of applications, where do we need to get to technologically to make this happen?
I think what we need is these measurements to be incredibly cheap, incredibly accessible. Because if we don't have those two ingredients, we'll just never be encouraged to measure enough, right? I think that's clear from the technology we're living now today. It's just so easily accessible to have a 12 megapixel camera. You take pictures of everything. I don't write anything down, I'll take a picture. I think if we have the tool where we can always measure a protein, we will always measure the protein. Even if you're not interested in the first instance, what the protein may be able to do or is doing in the system, we'll measure them.
Yeah.
And then we'll learn from it. And I think that's what we need. We need highly accessible, super cheap, powerful protein, detection technologies.
All right. Perfect. Thanks so much for joining us through this exciting conversation. As we've talked about, it feels like we are on the cusp of a tremendous breakthrough in proteomics that will have implications through biomedicine, through environmental studies, through food security, and beyond. Really delighted you were able to join us. Thank you.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.